The broadcast is now starting. All attendees are in listen-only mode.
Hello, hello. Welcome everyone to our new webinar from the Enology webinar series. It's great to have you all here. Very exciting. Again, a very international audience. Yay, thanks for taking the time to be with us today. Before I start, just a few words about housekeeping. As you probably know by now, there's a presentation that will take about 45-50 minutes and then the Q&A session at the end. Please type your questions in the Q&A box on your screen there, and we will do our very best to answer them all at the end of the presentation. Also, there will be a survey that will pop up once the webinar ends. I would very much appreciate it if you took 1 minute of your time to fill out that survey. It helps me tremendously in continuing this series, and it really only takes 1 minute of your time. So please do that at the end of the webinar. Also, if you have any suggestions for future topics for our webinars, please send them to me. I appreciate your input and your feedback about that. And now we should start our presentation today. We're going to be talking about nutrition, wine yeast nutrition, and driving wine style through that. Our presenter today is Mr. Luke Holcomb of Scott Labs. He is a recurring presence in our webinar series. I really appreciate you being here, Luke. I really appreciate you taking the time and all the information that you're sharing with us. And without further ado, let's begin the presentation.
All right, thank you. I have to reciprocate the appreciation. I really enjoy doing these. It's a great chance, especially in this day and age, for me to connect with people around the world and share some some of my experience, some of the things that I've learned and continue to learn. So I very much appreciate the opportunity. So, beyond completion, driving wine style through yeast nutrition. The basis of this presentation is given an assumption that in 2020 we do have the knowledge and expertise and the resources now to not be so concerned about getting primary fermentations completed. We can now move beyond that. So if you are having continual stuck fermentations on a regular basis, call me. We can work through it. There's a wealth of resources that I can point you to, that I can help with, a lot of that stuff. So that's the assumption that we're basing a lot of this on. So let's dive into it here. So the overview of what we're going to be talking about today. Sorry, little yeast over there on the right hand side of the screen. So what is YAN and how is it measured? We're going to talk briefly about the role of nutrients and also micronutrients and why those are important and how they function. We're also, like I said, going to talk about advancing beyond fermentation security. And specifically, we're going to look at the application of Scott Labs product Stimulus Chardonnay to improve aromatic intensity in Texas Rhone whites, because I think it's important that to not just talk about things globally, but talk about them specifically. And since this is Texas A&M, that's what we're going to talk about. However, this has application outside of Texas. The why is later on in the presentation, but essentially it's driving batch diversity in the cellar, and we'll talk about what that actually means. So what is YAN? YAN is yeast assimilable nitrogen. And that is the combination or the sum of your organic and inorganic nitrogen sources as well as peptides. There's a wealth of information out there if you want to dive deeper into all of the different amino acids and the ammonia and the peptides and the lipids and all that stuff. And we'll talk about it briefly, but there's plenty of information out there. Again, I can point you to those resources if you would like. We can have sideline conversation about it at a later date. But for the purposes of this presentation, I just want to establish the fundamentals that YAN is yeast assimilable nitrogen. how is it measured? In the yesteryear, formal titration, which involves formaldehyde and is somewhat inaccurate because it measures a few things that don't really matter, and there's inhalation issues and disposal issues with the formaldehyde. So most people are, and I think the rest of them should, move away from that. The current method that most people are employing with very good success is the NOPA method via enzymatic spectrophotometry. It's a fairly inexpensive, accurate, repeatable analysis once you get past the initial capital investment of the spectrophotometer. You can get lucky like I did about 10 years ago and scored an $800 spectrophotometer that was, retail cost about $6,000. But they've actually come down in cost quite a bit. There's refined systems out there that can help you to measure not only YAN, but also acetic acid and glucose fructose and malic acid and basically anything that can be degraded by an enzyme. It's a very powerful tool to have in your lab. And if I was setting up a lab, 100% required equipment, in my opinion. So what does YAN do? It is a key nutrient that determines yeast growth and fermentation rate. It is highly variable in grape must. It's essential to be measured, as I previously stated, and the ranges can be from almost 0 to 600. I've seen it very wildly in my own winemaking career. If I've seen it, for example, where fertigation was implemented on a very large scale vineyard operation, and the YAN levels were quite high. And it was actually a struggle to rein those fermentations in. They were so active. So high YAN is not necessarily a good thing. And low YAN, more often than not, is not a good thing either. But we have the tools in our toolbox to, at least in the case of low YAN, definitely supplement and control that somewhat. One of the issues with high YANs, just very briefly, depending on the profile, whether it's organic or inorganic, if it's really high levels of organic and there's residual left over after the primary fermentation and after the biological conversion of malic acid, if you so choose to do so, it can be left in there and be used as food for spoilage organisms. So if some is good, more is not necessarily better. So we'll talk about that a little bit more here in a second. It also strongly impacts wine composition and flavor. And what we're specifically talking about today is aroma synthesis. So this is why I wanted to present on this topic, because we as a community can move past fermentation security at this point. We understand it. We understand yeast physiology. We understand how to measure and compensate. Now we can get into tailoring wine style and quality. Also, yeasts vary wildly in their YAN requirements. So not all things are created equal in the yeast world. Between suppliers, within each supplier's portfolio, there's a lot of stuff that happens in the background there. But suffice to say that even in the Scott Labs portfolio, we have yeast strains that require very little and some that require a lot more. The other thing that is not on this presentation that I would like to just briefly touch on is microbial competition. So, for example, your YAN requirements, if, say, for example, you have a lot of botrytis in your vineyard, your YAN requirements are going to be much, much higher because that botrytis has consumed a lot of the YAN. So you're going to need to supplement even more. Also, when you do implant your yeast strain, your Saccharomyces, it's going to have to compete with the other indigenous microflora that's there. Because, if you have bird peck, or if you have hail damage, or if you have rot, remember that you're not just dealing with one specific organism, say botrytis. You're probably dealing with a host of spoilage organisms. You've got the You get the fungals, you got the yeast, you got the bacteria. You're going to have a cornucopia of stuff. Now, given the profile of chemistry and temperature, relative humidity, and all that stuff, usually one is going to be at a higher population. But one of the things that happens with microbial communities is that there is antagonism and there's synergisms, synergistic effects that occur, where, for example, Pediococcus will do one of the intermediary steps in the formation of volatile phenolics for Brettanomyces. They basically serve the volatile phenols up on a platter for Brett, and then Brett continues it on to the next step, going from ethyl to vinyl. So in that case, the point there is that there are many organisms at work, especially when you have spoilage. And in Texas, where you have many different growing regions and typically a long grape haul, that becomes very, very much more of an issue than if you were in an estate vineyard, for example. There's a whole other topic that I'd love to talk on at some point regarding that, and we'll cross that bridge at another point. However, in this case, what I want to point out here is Yan by itself is not enough. It may be enough to get that fermentation complete, but it may not be enough to get the quality and the diversity and the complexity that you want. So here on this slide, you have a few different things that we're going to look at when it comes to yeast metabolism and yeast health and the life cycle of the yeast and what we want these yeasts to do. So yeast will consume the glucose and the fructose by the way, with different preferential nature. Glucose is much preferred by just about everything on Earth over fructose, and it has to do with the structure of the fructose and what they have to do in order to metabolize it. But that's another topic. This primarily goes to form ethanol and also CO2. Okay. The nitrogen is essential for protein synthesis, sugar transport, biomass production, and CO2 release. So the way microbial cells work is they constantly have to pass things back and forth across their cell membrane, and this a lot of times requires energy. There's the difference between active and passive transport. So in order for that sort of thing to happen, you need nitrogen and some other things in order to aid that mechanism. And the healthier that cell wall is, the better the yeast will be able to do that. They don't have to divert resources as much if you're supplementing with these complex nutritional enological products like we're talking about today, like Go Firm Protect Evolution, Stimulus Chardonnay, Fermento, for example. And vitamins and minerals, these are important for enzymatic cofactors, which is fundamentally involved in aromas, as well as a host of other things like breaking down skins, right? If your yeast has good enzymatic activity, it will aid in the breakdown of polysaccharides and pectins and things like that. What we're focused on today is aromas. We'll talk about a little bit more here how enzymes affect that. And these can be endogenous, meaning enzymes that are naturally occurring or present in the beverage, for example, that which is derived from yeast, but also could be exogenous, meaning you add them in a refined format, something like our Scottzyme BG, which is beta-glucosidase. And then lipids. So these are essential for yeast survival. This greatly affects and limits the risk of stuck fermentations. Also, oxygen, super important. And if you know me, you understand I am passionate about this topic. In the case of yeast metabolism and fermentation health, it is vital to have proper oxygen. You can do this through something like Go Firm Protect Evolution, which is a rehydration nutrient. This is actually biologically adding some of the oxygen that you need. You can also do it with a sparging stone, for example, and inject gaseous oxygen right into the liquid. You can also use something like a Venturi. These have different applications for different styles of wine and how your cellar's set up. And if you want to talk about that, please reach out to me and we can go to a depth that you'll probably regret after talking to me, but it's my passion topic. So real quick, this is a point in the presentation where I'm going to point you to the resources again. Dr. Nicola Hall, who's our staff scientist on board at Scott Labs, is involved in a webinar series, and she's done 2 parts at this point, dealing with everything yeast, and goes to a much greater depth in the technical aspects and really delves into the science. And that's not my forte, that's hers. So they're available on our website and on our YouTube page, scottlab.com and Scott Laboratories on YouTube. But what I really want to focus on here is the acetyl-CoA, which is vital in the production of aromas, and it's an enzyme cofactor that is essential for enzyme activity and for yeast metabolism. So beyond YAN, having adequate YAN might get your fermentation finished, but might not support improvements in quality. For example, pantothenic acid, B5, B5, has a huge impact on H2S production. Pantothenate deficiency leads to high hydrogen sulfide. This is that rotten egg smell, even in the case of high YAN concentration. So you can see this little graph here. You notice that even in high YAN, 250 adequate YAN concentrations, you still have, you can have high H2S production if there's a deficiency in pantothenic acid. As you go from left to right, here you see the concentration in milligrams per liter, it looks like, or micrograms per liter, 10, 50, and 250 respectively in 2 different YAN environments, 60 milligrams and 250 milligrams. And you can notice that there is a wide disparity in H2S production. So you can have 250 parts of YAN, which is plenty sufficient to get that fermentation done, but you can still have a super high level of H2S, which is going to be a bear to deal with in the cellar. And this is why I'm saying we can move beyond some of these things. I mean, this research was done in 2003. So also example number 2 in the purpose of this presentation, for example, Chardonnay, Stimula Chardonnay. This is a yeast nutrient available from Scott Laboratories. And what you're seeing here is 4 different esters. And this was added at 40 grams per hectoliter at the first third of fermentation. And this references the increase in ester production when using this nutrient as opposed to DAP. So DAP is like 1960s technology on yeast nutrition. And we as an industry can move beyond that at this point. So I'm going to cover some of the aromatic compounds common in white and rosé wines. I'm leaving out reds. This is not the purpose of this presentation. We can talk about that at a later date. Terpenes. So these are floral aromas as an encompassing description, common in muscat varieties. So Muscat Canelli, Muscat Alexandra, Orange Muscat, Black Muscat, on and on and on. To a lesser extent in Riesling and Gewürztraminer. Glycosidases are involved in the liberation of the non-aromatic monoterpenes here. So this is where doing a bench trial on a finished wine with our Scottzyme BG can actually make a wine be more aromatic by liberating these terpenes that are bound to glucose. So that's why it's beta glucosidases. Fusel oils. These are higher alcohols produced by yeast during the fermentation. They're sometimes described as sweet smelling. Thiols, we're all very familiar with these in one way or another, whether we know it or not. These are sulfur-containing positive aromas common in Sauvignon Blanc. This is, the coup de grace for Sauvignon Blanc and why it smells the way it does. 4-MMP is boxwood, 3-MHA, and 3-MH, all are the passion fruit and grapefruit aromas. For this presentation, we're going to be focusing on esters. So these are fruity aromas, apple, pineapple, stone fruits, etc. Now, one thing I'd like to point out at this point is When I say fruity and I say something specific like pineapple, sometimes consumers and people think that we're adding pineapple juice or essence to the wine. We are not. This is actually the specific chemical that is responsible for the pineapple aroma. It's chemically identical, and it can be found in wine produced by yeast based on the substrates that they work off of and what they are able to, what they're enabled to do that winemakers control. So it's common in Chardonnay, also Marsanne, Roussanne, Viognier, which we're talking about today. There are also negative volatile sulfur compounds such as hydrogen sulfide, mercaptans, and disulfides. That's a very complex topic right there in of itself. Also, ethanol is very important for overall intensity and volatility. If you get really bored and want to do some alcohol trials, spike a wine with a few different levels of neutral grape spirits, let it sit for a while, and you'll notice that there's a great impact one way or the other, whether negative or positive, based on alcohol content. So, important takeaway here, the precursors for these aromas must be present in sufficient quantity, and the yeast must be able to liberate them. We'll talk about that here in a minute. This is one of the reasons why yan is not enough. You need the precursors in order for good aromas to be produced. So going beyond yan with your yeast choice, some of you might be familiar with this. This is available online. This is in our handbook. This is part of our yeast strain selection guide. But what I want to focus on, there's a lot of things you can look at: alcohol tolerance, nitrogen needs, temperature range, blah, blah, blah. But what I'm mostly concerned with, and the purpose of this presentation, is the sensory effect. So there's many different categories that we put these different yeasts in, but for this presentation, we're focused on esters. You can have things from a neutral sensory effect to esters with mouthfeel contribution, which is signified by the M. There's yeast strains that generally are enhanced varietal characteristics. That's the EVC. You have some that have effect with esters and varietal character, just varietal character esters with mouthfeel component, those are usually polysaccharides, and so on, and so on, and so on. So, for example, we're promoting ester formation in Texas Rhone whites. We're talking Marsanne, Roussanne, and Viognier through the use of stimulus Chardonnay yeast nutrient. We'll dive into that a little bit. First, you have to have a nutritional strategy. Okay, so you got to choose your yeast strain, and as I mentioned before, they have varying requirements for nitrogen. So you have to understand what that is and select the appropriate column, and then you have to reference your Brix as well as your microbial populations and some other things. But you need to determine your nitrogen requirements based on the parameters of your yeast and your fermentation conditions. Then you calculate the amount of nitrogen by measuring what's endogenous and subtracting that from the necessary amount of nitrogen, and that is going to give you the amount of yan required to supplement. Then you're going to determine what your goal is. If it's just fermentation security, that has a different program. Fermentation security with thiols being something you want to promote, or esters, for example, and we're focusing on esters today. So, esters, and you see the picture in the background, you got some citrusy, some strawberries, some berries, and things like that. These are the esters fruities that we talked about earlier. So in this case, you have, this is choosing fermentation security as well as ester production. So you have first column, yan required to supplement, and 0 to 50, 51 to 100, 101 to 150. And at the rehydration phase, you use that go from protect evolution to aid in sterol production and cell wall shoring and some other things to help the yeast later on in the fermentation. At 2 to 3 Brix drop, you add complex organic nutrients such as fermate O and then at 1/3 you add the stimulus Chardonnay, because it's important to have the right timing on this too, because the mechanisms behind aromatic production happen at different phases of the yeast life cycle and fermentation stage. So it's important when we say 1/3, we mean 1/3 of the sugar drop. Stimulus Chardonnay contains the aromatic precursors in the form of amino acids as well as the vitamins necessary to aid the yeast in the production of esters, which is why it's different than, say, Fermate O, which is also organic nutrition but has a different amino acid profile. These are the ones in Stimulus Chardonnay are specific for aromatic production of esters. So first phase, healthy yeast, then we have happy yeast, and finally we have the esters. So this looks like in Viognier, you got Viognier, select a yeast strain such as B fruits, fruit being esters, and combine it with Stimulus Chardonnay. So that's kind of a pretty basic recipe, if you want to call it that, for maximizing ester production. And there's also something else to be said for specific fermentation tannins, which can increase the pool of those precursors for aromatics, as well as something such as glutastar, which is contains a lot of glutathione, which will help protect your aromatics from oxidation and also help keep the color of your white wines closer to the yellow side as opposed to the gold side, which Viognier and some of the Rhone whites are prone to going more gold. So if you're wanting to keep it more on the crisper yellower side, glutastar is a good thing to look at. So the impact of nitrogen on aroma, this was done at OSU, Oregon State University. This was on a Chardonnay. This was done analytically. I'm not sure the method, whether it was HPLC or GCMS, but it was some sort of chromatography. But using just DAP for the fermentation nutrient as opposed to Stimulus Chardonnay, you can see some of the relative increases in concentration here. These are odor-active, so when you look at data on a slide or in a presentation, you know, sometimes it's like it might be measurable, but it might not be effective. It might not have an impact. This specifically is looking at actual impactful aromas here, okay? And these are categorized less specifically, so they're kind of in combination because these esters work synergistically in the aromatic perception, right? So the sum is, the whole is greater than the sum, right? So it's not just any individual ones. As you increase the pool, the perception increases. So why would you drive style one way or another with these tools? So blending diversity, different programs, and just good old-fashioned experimentation. So the concept is, you know, maybe you've got a lot of Viognier that's contracted, for example, and you don't want to make just one batch that is neutral, that is the same old in all situations, but you want to have diversity from a blending standpoint, creating different programs, having some complexity built in there. This is why you would split it into a few different lots, and you can drive those lots apart from one another stylistically using the tools that we're talking about today: yeasts, nutrients, fermentation tannins, things like glutastar to do so. So you can push things diolic over into esters based on the nutrition that you feed it. You can also potentially make a neutral wine if so desired, right? This is just about diversity and driving things in different directions on purpose. So for example, real quick, enhancing thiol production in Viognier would look like Viognier plus Sauvy, which is one of our newer strains, which is really high thiolic production. And then you would combine that with Stimulus Sauv Blanc, for example. And so, you know, in example one, you're taking things in the ester direction. Example two here, you would be driving things into the thiolic region. And again, you can use the fermentation tannins and glutastar. Example three would be making a neutral Viognier, for example, using something like DV10, which is a phenomenal fermenter. It's just not very diverse in its aromatic production, right? It's fairly linear and one dimensional. And then you use Ferment O, which while compared to DAP will most of the time make a more aromatic and pleasurable wine. It won't to the extent that the stimulant products can do. So real quick, here's a look at the nutrient impact on Gruner Veltliner, which, I think there is a decent amount in Texas. So you have the sum of the esters and acetates here. The first column, this is in micrograms per liter, nearly 12,000 being produced with Stimula Chardonnay or Stimula Sauv Blanc, about 11,000, and then less in the control. These things are actually in the control is Fermento in this case. And these concentrations, these are odor active. You can tell a difference. Now that is definitely dependent on your own genetic capability that's baked in from the factory. Some people range in their ability to smell these things, right? I may smell something and tell you, oh, that has an increased guava compared to the other wine, and some people will be like, yeah, it's a white wine. I don't know, and that's just how we're wired, and you can train that genetic ability or lack thereof to a certain extent, but you kind of have to come equipped from the factory. So moving on, in the 3MH and 3MHA, those are thiols again, and you notice the middle bar is correlated to Stimula Sauv Blanc, and you can compare that to the other two nutrients, Fermento and Stimula Chardonnay, that have much less thiols. So this is just showing you that there is an impact and you can drive wine style with nutrition. Harvest 2020, we are trialing Stimula Syrah and Stimula Cabernet for red wines. It's not in a handbook, but it is somewhat available. Please contact me if you would like to set up a trial this year. So photo credit here, and basically credit completely because Nicola Hall is, Dr. Hall is like my master Jedi, and I'm the young Padawan. But photo credit goes to her. This is Cabernet Sauvignon in Rutherford AVA. Vineyard is George III, some of you may be aware. So in summary, we get our vitamins and minerals and sterols from Go From Protect Evolution. We get our nitrogen from things like Ferment O, and we get our odor active amino acids from the stimulant products. So that's it in a nutshell. I think we're right at about an hour, so that kind of worked out. Thank you for your time. And there's my contact information, my email, my Instagram, should you choose to do the gram stuff and follow me. There's also my cell phone if you would like to call and talk. And then all of the Scott Labs material on the socials as well. So that's it.
Well, thank you, Luke, very much for this presentation. Now, if you guys listening in have questions for Luke about this, please type them in the Q&A box there, and I will read them and we'll try to answer them. Luke, I had a question. If you could go back on your slides to that table where you had the intervals for adding yan and then 3 different other columns.
With a lot of clicks, but
no.
That one right there?
Okay, yes. So can you, I'm sorry, I'm probably missed this, but what does the first column mean, yan required to supplement?
So that's when you have analyzed your fermentation medium.
Yeah.
And discovered how much endogenous yan there is.
Okay.
And then you factor in how much is needed to properly get the fermentation complete. And this is based on microbial diversity, pressure, basically. This is also determined by sugar content, aka potential alcohol, and then also your yeast strain that you've chosen, right? So once you look at all those factors, that can tell you how much yan is required to supplement in order for fermentation security. So that's the first column.
Mm-hmm.
second column at yeast rehydration phase, that's when you're preparing the active dried yeast, you use a rehydration nutrient such as Go From Protect Evolution to give them the tools necessary to finish a fermentation properly, mostly through sterols and ergosterols, which allow for cell wall fluidity, which helps at the later parts of the fermentation when the alcohol is high. And then you have your two other columns where you're adding, say, something like ferment O to hit your necessary YAN requirement that we determined previously. That's for happy yeast. That's to make sure that they're going to do what they need to do, just from a strict, alcohol production standpoint and the consumption of sugar. And then in the case of stimulus Chardonnay, at 1/3 of sugar depletion, that's when you add it, and that's going to help. That's the point at which, and that's the product you use to increase or optimize the production of esters in your fermentation medium. In the case of this presentation, Rhone whites.
Right. I understood all the other columns. I wasn't sure how to interpret the first one. So really, based on the YAN required to supplement, the only thing that varies is the ferment O addition.
Correct. And you can go to our website. We have all of this online there. And I just pulled up the slide that determines that first column, right? So it's the sugar content, it's the yeast strain, and then, some other things like, is it going to be a stressful environment, right? Are you going to ferment really cold? Are you going to ferment really hot? Did you have rot in the fruit? There's a host of, do you have temperature control or not? There's a lot of other things that you need to look at when deciding how much nitrogen you're actually going to need. Okay, so this gets into yeast strain selection a lot. And again, I would point you to Dr. Hall's presentations on that are available on our webpage and our YouTube channel.
Well, this is very interesting, and I think very helpful as well to have all that information, and it might warrant a webinar on its own.
Oh yeah, yeah. That's why, like I said, the premise of this whole presentation is not to talk about fermentation security. I feel at this day and age, there's enough content out there. There's enough resources, people like you, people like me, other people at Scott Labs, that can help you get to a point where you don't have sluggish fermentations anymore. We have the tools to do that. This presentation was like, well, okay, since we've moved past that, how can we drive quality and drive blending diversity and drive different aromatic production and things like that? We can even get into like mouthfeel and color and a whole bunch of other stuff that we could get into once we've moved past nutritional fermentation security.
Right. We have a number of questions actually coming up. So the first one comes from Hector, and the question is it recommended to use a complete nutrient at the beginning of fermentation and then use only DAP?
So that may be the recommendation from the 1990s. I'm directing almost every one of my customers in most scenarios to get away from using DAP. Again, DAP is pretty old technology. It's very cheap, but while it will help you get your fermentation done, it has some negative implications insofar as mouthfeel and aromas. It actually shuts off a lot of the aromatic pathways inside the yeast cell. So you get, generally speaking, if you compared organic supplementation, like what we're talking about in this presentation, to strictly DAP supplementation, the wines are going to be much higher quality with organic nutrition. The DAP are going to be harsher, less aromatically intense. On the mouthfeel, they're going to be, again, a little harsher. So you can use DAP, like I said, but there are some drawbacks to it.
Thank you. Next question comes from Marek. Is this presentation going to be available somewhere in the archive? The answer is yes. I am recording this presentation. I will post it on my YouTube channel. And so, yes, it will be. Next question. What are the effects of YAN more or less quantity on sparkling method traditional secondary fermentation wine? Does it affect aroma profile?
Yeah, that's the short answer. That's a whole nother rabbit hole, super complicated black art kind of subject when you start talking secondary fermentation, whether it be Charmat or method champenoise. It again, that's a whole nother rabbit hole that I'm probably not the best person to ask that question of. I'd rather point you to some of the resources we have internally for that specific question. And I'm happy to do so. Just contact me and I'll put you in touch with the people that know that answer better than I.
A question from Bob. Any thoughts on Blanc du Bois with the use of Stimula Chardonnay, since Blanc du Bois has some muscat in its genetics?
Yeah, actually have a very well-known winery in Texas using these products on Blanc du Bois and having very good success. So, I don't have at this exact time some graphs or tables or data to show you from Blanc du Bois, but I do have anecdotal proof that they work on Blanc du Bois.
Thank you. That's great to hear, actually. Very exciting. Next question from Rennie. In the same slide of the first question, so I assume the slide that I talked about, is there any specific reason that you mentioned a 2 to 3 Brix drop for an addition and then mentioned 2/3 sugar drop? Are these not the same value?
No, they are not the same value, and it has to do with the life cycle of the yeast and what they're doing at that point in the fermentation process. That's why it's specific. At 2 to 3 Brix, there's certain things the yeast are doing that, for example, 2 to 3 Brix, they are in their exponential growth phase. They are budding, they are proliferating, they're increasing their biomass significantly, and they need the nitrogen at that point. They're multiplying, and that's when you want to add the nitrogen. That's why you add the fermate O at that specific time. So at 1/3 sugar completion, not 2/3, at 1/3 sugar drop, that's when you want to add the stimulus Chardonnay to drive the production of the esters, because that's what the yeasts are doing at that time. They're doing their primary, right? So primary metabolites would be heat, alcohol, and CO2. Secondary metabolites are these things like aromas and esters and terpenes and on and on and on. So at 1/3 drop during an active fermentation, that's when you want to supplement with the specific nutrient that contains those precursors to drive the production of those esters or thiols if you're doing that.
Yeah. Thank you. Next question. We use DV10 or D21 yeast for sparkling second fermentation. How can we gain more aroma profile after second fermentation, provided 24 to 36 months yeast aging and using stimula instead of DAP?
Yeah, again, I wish I could answer that. I can nibble around at the edges of that, but I would really prefer to answer that offline.
Mm-hmm.
Sparkling is just a very complicated process that has other concerns that I don't think is reasonable to address at this point.
Mm-hmm. And the last question so far, so if you guys have any more questions, please type them in. What is the difference in red and white grapes for nitrogen adding?
The answer is it depends. There's a lot of differences, but at the same time, there's not. This is the dichotomy of everything that we do in life, and in winemaking, we're striving for balance. And as you change one factor, it has effect elsewhere. I like to picture this in my brain as a spider web, and when you put pressure or pull on one area of the spider web, everything else kind of morphs along with it, right? So things in nature and things in life don't exist in a linear fashion. They are dynamic. These are dynamic, multi-dimensional models that we're dealing with here. So with white wines, you process them completely different from red wines. So one of the things I'll point out here that is a big factor and has a big effect is solids. So commonly with white wines, what we do is we will crush them up, we'll press them in a press. Hopefully that's a Wilmus press, by the way. Little plug there. And we will then usually add a fining aid, perhaps, or maybe an enzyme, maybe some SO2. And what we're going to try to do is we're going to try to reduce the solids, commonly through cold settling. Usually 18 to 24 hours. There's other methods, centrifuges and things like that. But commonly cold settling, and then we rack off, and then we begin our fermentation. So the solids content of white wines are vastly different than red wines, where there's their skins and seeds and maybe some and pulp and stems and all sorts of other things in there. And so the solids content is drastically different. So for example, if you over clarify your white wine, you can reduce the solids content, aka as correlated to the amount of nitrogen that's left in there. You can over clarify it to the point where you actually get a stuck fermentation. And so if you have super, super highly clarified juice, most of the time we recommend supplementing with yeast hulls and fermentation nutrients in order to overcome that lack of solids. Whereas in red wine, you have all those solids. So that was a lot of words to convey a pretty simple concept.
Yes, but it was a very complete answer.
Okay, good. But so just to dovetail off that for those people that are still subjecting themselves to the sound of my voice, but I think one of the concepts that has really describes what winemaking is segregation, it's clarification, it's solids management. I think that is one of the least talked about but most important thing in winemaking, and I'll describe it briefly. You separate the grapes from the vines, and then you separate the grapes from the stem, and then you separate the juice from the grapes, and then you separate the colloidal solids material from the liquid, and then you remove the microbial populations out of that liquid, then you have wine. And if you do it correctly, you have shelf-stable wine.
I never thought of it that way, but I like it a lot. Now that I heard it, I'll keep thinking about it like that. Yeah. Yeah. All right. Well, we have no more questions at this point. So if nobody else has a question, then we will conclude here. Look, thank you so much again for a really informative presentation. Thank you all for being here. Please take a minute to fill out the survey that will pop up. Oh, here we go. Your dogs are very good boys and deserve extra hugs.
Thank you. I wore them out this morning with a long run, so that's why a good dog is a tired dog. And some of you might know Mr. Wilson. He's my male dog, and then my newest addition is Miss Luna back there. She's a full-blood 4-month-old German Shepherd, so I have my hands full. But thank you for the compliment.
All right, thanks again to all of you, and stay safe, stay healthy, and we'll see you all next time. Bye-bye.
Thank you.